next we are supposed to study about TS of dicot stem. In the dicot stem TS, it reveals the presence of three types of tissue systems here. Those are epidermal tissue system, ground tissue system and the vascular tissue system. Now here epidermis, in this the first and the foremost layer will be the epidermal hairs which arise from the epidermis. Next one, this is the circular outline of the TS of dicot stem. So in this first and the foremost layer we are supposed to study about epidermis. What is epidermis here? As we all know, it is a protective in function and it forms the outermost layer of the stem, and it is generally made up of single layer of cells which appear almost rectangular in transverse section. Now, these cells are compactly arranged and do not possess any sort of intercellular spaces. The cells are living and are capable of undergoing mitotic divisions in the radial plane and also undergo tangential enlargement. Now, next one is, it is also epidermis is also considered as the made up. The outer walls of the epidermis are consisting of highly cutinized walls, and it consists of waxy coats and hairy coverings are occurring in many cases. Presence of cuticle and the presence of waxy substances is almost present, and it is almost act as a waterproof layer, so that very little amount of water is lost by transpiration because of the presence of cuticle and because of presence of some waxy substances. And as we say, we have a labeling there called as epidermal hairs. So these hairs are nothing but these are the outgrowths of the epidermal cells, as I said earlier, and may be present sparingly or such, or in some members in certain plants, to give rise to a stem, a woolly like appearances in the structure, and stomata is present to a limited extent in the epidermis of the stem. Next we are talking about hypodermis, and what is hypodermis here we will talk. In this regard, first we are supposed to talk about cortex. The cortex, next layer to the epidermis, is a few to several layered thick zone of cells called as cortex. This region can be differentiated into two zones, of which one of this zone is collenchymatous hypodermis and is made up of varying layers of cells and lies immediately beneath the epidermis. It is immediately beneath the epidermis, and normally in, for example, as per your syllabus, you can take an example of cucurbita stem in which the hypodermis is well developed, and in hypodermis below the ridges, the curves which are there below the ridges is a single layered and is absent elsewhere. It is so hypodermis is present below the layer in a single layer. The cells have localized thickenings and may possess chloroplast, undergoes several dual functions like of protection and also of photosynthesis. In many dicots like Fumaria, you can see the presence of collenchymatous hypodermis, and the remaining cortex and the entire cortex is parenchymatous. So hypodermis here is collenchymatous and the remaining cortex is parenchymatous. The next layer immediately to the hypodermis is called as general cortex, or we can also call it as the median part of the cortex. The parenchymatous layer, actually it is nothing but the median part of the cortex or general cortex. Next, it consists of loosely packed parenchymatous cells, most of which contain chloroplasts. There are prominent intercellular spaces present in this part of the cortex. The cells are generally large, rounded, oval and sometimes polygonal in shape. Okay, for example, in your syllabus, as we take an example of sunflower stem, you can see the presence of isolated resin ducts lined with secretory cells are seen in the part of the cortex. So what we can see here, we can see the presence of some isolated resin ducts lined with some secretory cells which is present in some parts of the cortex. Okay, now next one, we can also see the presence of some sclerites in some plants, in presence of laticifers present in that. All these are possibilities which can be seen in the cortex. So next layer we are supposed to study is here, that is endodermis. What is the endodermis? Here, as we know earlier, it is the innermost layer of the cortex which delimits it from the stellar region or from the stele. It is called as endodermis. Now what is, as we already know, the cells of the endodermal cells are barrel shaped, and what is the function of endodermis here? It is helpful in the accumulation of the starch. So this is also recognized as a starch sheath. Majority of dicots are lacking a morphological differentiation between differentiated endodermis. It is impossible or difficult, we can say, to differentiate between a layer of cortex and a vascular lesion. So here this endodermis is also called as a starch sheath. Now next layer we are supposed to study here is pericycle. What is pericycle? The term pericycle is given to a layer or layer of cells that occur between the endodermis and the vascular cylinder. And the pericycle occurs in a continuously circular cylinder around the vascular region. In cucurbita and in some other members of Cucurbitaceae, you can see the presence of sclerenchymatous pericycle occurring in the form of a continuous cylinder made up of several layers of cells. So basically what we say, in Cucurbitaceae we will see a complete ring of cylinder, circular cylinder of pericycle. Now we are supposed to talk about here one word called as hard bast. What is hard bast? For example, in Helianthus it forms a alternating patches of thick wall. For example, if you take an example of sunflower, you will see the alternating patches of thick and thin wall cells present in the pericycle. So the thick wall patches which are occurring above the phloem, then those are called as hard bast. The thin wall patches are above the medullary rays and scarcely distinguished from the cells of the ray. So this is about your pericycle. Next we will be studying about vascular bundles, and before vascular bundles let us talk about medullary rays. What are medullary rays? Medullary rays are nothing but the parenchymatous area present between the two adjacent vascular bundle. As the indication itself shows in the diagram, in between the two vascular bundle a parenchymatous area is present which is called as medullary rays, and the cells of the medullary rays are all polygonal or radially elongated. They extend outward from the pith and provides pathways for, what is the function of medullary rays here? It provides pathway for radial conduction of food and water at any level in the stem. So now when we talk about vascular bundle, these all are medullary rays. Next is we will be talking about pith here. As we know what is pith, it is the central portion of the ground tissue and is extensively developed in, for example, we can call it as in sunflower, it is very good pith development. In this, the pith consists of large thin wall cells in the center, and the cells have intercellular spaces. The cells of the pith near the vascular bundles are comparatively smaller, thus distinguishing the peripheral portion of the pith from the central part. So towards the vascular bundle it is smaller, and towards the center they are bigger in cells. Now next we have left over with the vascular bundles. Now what is vascular bundle here? These vascular bundles are differentiated, as we know earlier, that it is from the procambium, which in turn develops from the derivatives. Where does this procambium comes? It comes from the derivatives of the apical meristem. So here the dicot stem, the vascular bundle arrangements are discrete and are arranged in the form of a ring. When we go through the diagram, you will see xylem and phloem. See all the vascular bundles here, red, blue and a green color line in between them. Here the vascular bundles are discrete and arranged in the form of a ring. Now here, what is vascular bundle? Each vascular bundle is composed of two type of tissues, those are phloem and xylem. Okay. Now in this, if you see clearly the diagram where the indication of vascular bundle is there, in between vascular bundles, two vascular bundles, there is a strip of meristematic cells called as vascular cambium. If you clearly see, there is a green color strip going across each vascular bundle, that is called as vascular cambium. The arrangement of phloem and the xylem is collateral, and with phloem towards outside and xylem towards the pith. So this, because there is a presence of vascular cambium, we can say that this vascular bundle is open type of vascular bundle. So this is the total structure here. When we see the TS section, this was the circular section, this is the total TS section here. Now in cambium, when we are talking about the there is a vascular cambium in between the vascular bundle. So here it is nothing but a thin strip of cells extending lengthwise between the xylem and phloem. It consists of single layer of meristematic cells which appear almost rectangular in transverse section. It is just useful for the secondary growth. So this will be the TS section of the dicot stem. Till now we were seeing about circular section, like we have epidermis, outermost protective layer, next collenchymatous hypodermis, next parenchymatous general cortex is present, or median part of the cortex, as I said earlier. Next you have endodermis. Next we will be talking about pericycle. Next we said we have phloem, next cambium, next metaxylem and protoxylem elements are there. Next we will see the presence of pith. So, these are the details of TS of dicot stem. The vascular bundles of dicot stems are normally collateral, conjoint and open with endark xylem. So, this is about TS of dicot stem.